Good day. It's a pleasure to be with you today. Thanks to Mauro Schechter and Susan Buckbinder for inviting me to this meeting. And my goal is to sort of give you an overview of where we are in HIV vaccines in 2021. 2021 is a pivotal year for us in HIV vaccine field. In 2015, the HIV Vaccine Trials Network really set out a conscious strategy to execute a series of efficacy trials to determine whether its non-neutralizing antibodies could be elicited at a high enough titer to be clinically useful as a follow-up, say, to the RV144 trial, or to determine whether we could prevent acquisition with broadly neutralizing antibodies as a model for BNAB-inducing vaccines. Now, some of the answers have come in 2020, and this year will sort of complete the task. So we had a fork in a road. We did two trials that are non-neutralizing antibody trials of vaccines, HVTN 702 and HVTN 705. And then we did the passive immunoprophylaxis studies, what we call the antibody-mediated prevention trials. In 2015, in January, we started this task of improving upon the results of RV144. It is the trial in which Alvac and AIDS-Vax were used to prevent HIV acquisition in Thailand. It had early a 60% efficacy, overall a 31% efficacy. And from that came the initiative called the P5 initiative that at that time had Novartis, the military research program, us and the HVTN, the Gates Foundation, Sanofi Pasteur, South African Medical Research Council, and the National Institute of Allergy and Infectious Disease to build a public-private partnership to actually reconstruct the Alvac kind of vaccine into a clade C format and test this and hopefully use it to improve upon RV144. The strategy was really multifocal, but the one that went ahead the most was to use an Alvac clade C, use bivalent clade C GP120, add the adjuvant MF59, add a 6 and a 12-month boost to the program, and see if we could improve upon the immunogenicity of the results. We also then did a non-neutralizing antibody approach with the Ad26 profile. And the reason we did that is that the Ad26 vaccine really had a very clean profile and highly protective vaccine in non-human primates. It was built on a mosaic antigen, which is a synthetic antigen that had eliciting T-cell epitopes, and it turned out even B-cell epitopes to non-neutralizing antibodies with greater clade breadth than in the 702. The correlates of protection had some overlap between the two regimens, but there were many differences, especially in the breadth of both binding and T-cell responses. And the diversity of the immune responses would improve the ability to define what non-neutralizing immune responses are correlated and what areas of the viral envelope are susceptible to clinically effective immune pressure. This sort of spider plot actually summarizes a lot of data that was done in the qualifying trials, HVTN 100, which used the Alvac bivalent GP120, and HVTN 117 that used the Mosaic Ad26 with the GP140 boost. The slide shows two things. Anything that had to do with GP41, like out here, always favored the Ad26 regimen. If we go around to the sort of V2 loop area of things, you see that the Alvac regimen is a little bit better. And anything that actually had to CD8 T-cell responses was elicited by Ad26 and not by the Hox protein, the Alvac regimen. And both the cytokine responses and the breadth were also different in the CD4 responses. Sort of illustrated in this slide that looks at the sort of polyfunctionality and the immune response of the CD4 T-cell responses between HVTN 100, the Alvac, and HVTN 117. And you see it's a much more sort of cytolytic pattern from the adenovirus vector. Here's multifunctional cells making interferon gamma, TNF, as well as granzyme B. Favoring the Ad26 regimen. And then here in the CD8 T-cell responses, you see much greater CD8 T-cell responses in the red, which is 117 as compared to the HVTN 100 regimen. The breadth of the response is also the number of epitopes is markedly better with the mosaic antigen. When we did the Ad5 vector, the median number of epitopes was only 2 to 3, and here you have much greater breadth in the teens, basically. Now, late in 2020, the HVTN 7002, even though beautifully done study of 5,400 people, recently published in the New England Journal. Glenn DeGray is the lead author, but it was disappointing. We didn't have any efficacy whatsoever. The curves were right on top of each other, whether that's force of infection or the fact that when we reconstructed the vaccines, they're not exactly identical to the ALVAC regimen as it relates to the V2 loop. Our studies are starting to determine and undergo whether that is the case. Just to remind us, main correlate out of RV144 was this antibodies to the conserved region of V2 were highly correlated with efficacy, and this is the sort of the higher the titer of V2 antibody levels, the higher the efficacy. This is based on RV144. When we did the qualifying trial for the ALVAC regimen, we actually showed that binding antibodies and T-cell responses, both in prevalence and magnitude, were actually much higher with the regimen that we constructed for the South African trial for 702 than from RV144, or in HVTN 097, which we used the RV144 regimen in persons who were in South Africa. But when we reconstructed it, even though the binding antibodies were better, the V2 loop responses here were better in HVTN 097. So this is the RV144 regimen using the A244 isolate as compared to the HVTN 100. If I showed you binding antibodies, the green would be much higher than the purple. But when it comes to V2 loop antibodies, the purple tend to be higher than the green here. And whether that was significant or not, whether A244 is a very special immunogen that actually creates useful antibodies is something we'll have to explore. When you look at the Ad26 regimen, recognize that it's a mixture of 4 different adenoviruses, 2 synthetic mixtures of GAG-POL and 2 synthetic mixtures of the envelope associated with a GP140 boost. That is given in a 0, 3, 6, and 12-month regimen. Peak titers really are after the 12-month regimen, about 25% higher than after 6, with excellent durability. This trial is fully enrolled and actually done being reviewed by the DSMB, and we will essentially analyze this trial in the end of July. And this will be an important milestone with respect to finding whether this vaccine holds any efficacy. We're seeing essentially the full cohort finishing its end of 2-year follow-up at the end of this month. The next trial that we then did was the antibody-mediated prevention trials. This was a series of test of concept trials in which we were giving a broadly neutralizing antibody to the CD4 binding site called VRC01. It was a trial that we first conceived in October of 2014, had a face-to-face by March, had a protocol development and face-to-face meeting in May, and really had our first patient in between April of 2016. So from concept to really getting into the field, a efficacy study of 2,700 persons of women heterosexual women in sub-Saharan Africa and MSM and transgender persons in the United States and in South America. So they were called separate trials, but were totally harmonized, differing in their patient population, but completely harmonized with respect to the operational aspects of things. VRC01 was given every 8 weeks by IV infusion for 10 infusions, so over a 20-month period of time. And we had incredible safety. We had an incredible trial in which we gave 45,000 IV infusions, essentially without incident. The safety of the humanized antibodies are really impressive. And as a test of concept trial, We analyzed the data in late August and published the data that came out in the New England Journal a couple months ago. The top line results are illustrated right here, which is that when the viruses were sensitive to VRC, we had high-grade efficacy. This is actually looking at the prevention efficacy. So this blue line here shows high prevention efficacy, 75 to 80%. And it is in the light blue with isolates that were sensitive at less than 1 microgram per ml. If the isolates were more resistant, anywhere above 1 microgram per ml, you did not see efficacy. It was interesting that whether we gave the lower dose, which was 10 milligrams per kilogram every 8 weeks for the 10 injections, or 30 milligrams per kilogram, the results were the same. So that's the good news. The less good news is that when you look at the number of circulating strains, only 30% of the strains were sensitive at less than 1 microgram. We had actually predicted that the antibody concentrations or the viruses would be sensitive in the less than 10 microgram range, which would have covered 60% of the strains from our previous panels. But so this is sort of like having one drug as compared to multiple drugs, sort of like AZT. Yes, if we have exquisitely sensitive viruses, we have high-grade efficacy. If they're more resistant, they break through. We sort of looks like we have a little bit of transient efficacy, but they eventually break through. So what this means for us is that we probably need to develop, or we do need to develop, sort of multiple binding antibodies, multiple broad and neutralizing antibodies, develop combination therapy for prevention. The other important issue that came out of this trial was we essentially validated the TZMDL assay, the pseudovirus neutralization assay that measures viral isolation sensitivity to antibodies or to antibodies, that allows us to sort of therefore calculate what combination of antibodies do we need. And the other aspect of this is that, as shown in this slide, we had a number of non-human primate studies done with multiple antibodies, a large publication that was shown on the prior slide that looked at over 250 macaques in the challenge models, mucosal high-dose challenge models used for these antibodies. And basically what we show is that the prevention efficacy as it relates to the ID50 is pretty similar in the non-human primate models, which is the blue line, as compared to the human line. And, you know, we need to get an ID50 of around 1 to 200, 1 to 250 to get high-grade efficacy. So we know we have combinations of antibodies. Not only we've improved upon VRC01 to have modifications of that antibody to give better concentrations, longer durability, greater activity. And then we combine it with monoclonal antibodies to the V2 loop and the V3 loop. And so we have combinations that we know that we can achieve these kinds of titers and have over 90% coverage. And that is where this field is going. Now, the use of antibodies has also helped us design approaches to HIV vaccines that elicit broadly neutralizing antibodies. We now know that if you give high titers and you achieve high titers of broadly neutralizing antibodies, we can get high-grade protection. So can we make vaccines that give you broadly neutralizing antibodies? And the last 5 years have really developed really an enormous leapfrog in the science as it relating to HIV vaccine development. A lot of this has been to develop trimers that can be used to actually elicit broadly neutralizing antibodies. Some of it is the concept that we need to actually sort of prime the immune system in a way that, in order to achieve broadly neutralizing antibodies, elicits a group of B cells that are sort of immature B cells that have some unique mutations that are required in order to lead to the sort of further driving of the antibody to where it achieves broadly neutralizing approaches. And we call those germline targeting or lineage-based vaccine designs, in which we take people with broadly neutralizing antibodies and sort of use the isolates that they have derived to actually see if we can more universalize them among everybody. And then we've defined some reasons like the fusion peptide or the what we call the EMPER region to develop immunogens that are sort of germline agnostic, but we can get binding antibodies. And we're using different kinds of technologies, nanoparticles, viral vectors, but stabilized trimers, and most importantly, from learning from the COVID world, the use of mRNA to help iterate these programs more closely. So the point is that we have multiple targets where the neutralizings are. The CD4 binding site we already know is at VRC01, but the V3 glycan, the fusion protein down here, the V2 glycan, all these areas of where broadly neutralizing antibodies exist. And the goal of the vaccine is basically to see if we can prime and stimulate sort of so that we at least get 2 epitope lineages of broadly neutralizing antibodies. The leading one is the CD4 binding site, but we also have the fusion domain or the glycans to work on. There are multiple CD4 binding site broadly neutralizing antibodies. They attack the virus in different levels. It's sort of nicely shown here between the purple, I guess the brownish and the yellow. You can see the antibodies sort of attack the CD4 binding site at different locations. They come from different lineages. And then the goal is how do you get a priming immunogen to sort of get a VRC01 lineage versus a B12 lineage, or what we call the CH103 lineages. And all these sort of priming regimens are in development to have clinical trials to sort of see if we can increase the B cell precursor pool to lead to these lineages of CD4 binding site antibodies. One of them here is developed by Leo Stomatatis that again is a VRC01 class 4 binding targeted antigen that goes to a different cell surface part of the virus, and the hope is to prime that germline B cell. Now, one of the things that we've learned is that how we give the antigen seems to influence the ability to recruit rare B cells of interest in the germinal center. And Shane Crotty has pioneered this both in mice and in non-human primates. We're starting it now in humans, in which you actually give the same amount of antigen. Let's say here would be 100 micrograms of, let's say, the Stomatatis priming regimen. And instead of giving 100 micrograms of one dose each time from the way we normally give it, you extend it out over a 20-day period of time. And from low antigen, we start with day 0 would be 2, 5, 10, 15, 20, and 48 micrograms. So over a 6-time injection period of time, you give 100 milligrams. And the reason for that is that it seems to alter the dance between the T cells and the B cells in the germinal center. And it gives a chance for these sort of low affinity antibodies to actually mature, being picked out and selected for essentially proliferation in the germinal center. And you give more, you seem to have high avidity, and the high avidity antibody is actually doesn't get these unusual mutations that you need to have to then have in the B cell to then take off and mature it more to be a broadly neutralizing antibody. Again, another new technology is mRNA technology. It makes COVID trimers, the full-length spike. Here we have a trimer that has a pre-fusion stabilized soluble spike. We have a membrane spike, doesn't have the back end of the trimer, which we actually know can give distracting antibodies. And then we're also going to make a trimer in which we knock out the CD4 binding site, attempting to, again, get antibodies. It opens up the glycans and allows some antibodies that were to the other part of the CD4 binding site that actually might, we think, elicit higher VRC01 lineages. And so that in one easy trial, we can iterate 3 separate different trimeric substances. And again, illustrating the power of mRNA to be able to allow us to iterate. One of the last things to talk about is the fusion protein. This is sort of the area after the virus lands. It changes configuration and it has a fusion state. And Peter Kwong has made a fusion protein that makes antibodies to the fusion state, given it to non-human primates. About 40% develop enough to actually prevent HIV acquisition in a challenge model. We're looking at boosting it with a trimeric complex. This is sort of germline agnostic, so it allows you to take all of us, and without having to do this sort of selecting out of these unusual mutational pre-B cells. We can take the whole antigen and treat it similarly to a vaccine and see if that kind of approach will get us to where we want. Lastly has been the use of using infants as a way of both as a model and to see if we can develop neutralizing antibody vaccines for maternal fetal transmission. The study led by Glenda Gray. The concept is that infants have more naive B cells, and they will actually develop these BNAbs both easier, more quickly, and in higher prevalence than adults. And so we're taking some of the regimens. This is Bart Haynes' regimen that looks at the GT120 lineage-based antibody that we've been using in adults in HVTN 115 and giving it to children. During the at the moment, 10 children are enrolled in this trial, and we'll actually see whether we are essentially conceptually able with more naive B cells to program the development of B cell precursors and neutralizing antibodies more effectively in infants than we in adults, and hence we would have a strategy with our current technologies to use vaccination to prevent breast milk transmission from mother to infant. And lastly, to say that T-cell based vaccines are still being tested, especially to conserved regions, whether it's an ad vector or DNA, or a chimp ad vector, or Lewis Picker's CMV vector. Not enough time to go through that at the moment. So the pipeline is rich, and the past 3 years have created an understanding that getting immunogens into clinical trials is needed. We need to pick up this understanding and iterate these trials quickly enough to create a momentum to come forward with a vaccine regimen of scientific plausibility and interest. We need to create a cadence similar to what has happened with COVID-19 vaccines. And this is sort of what I call the comparison of the need to iterate. The two left lines going straight up are what the CoVPN, what we did as an academic network in our 24 sites for the Moderna and Novavax trial. You can see we enrolled 6,000 people in an 8-week period of time. And the bottom lines are what we did in our efficacy trials, which really to get 6,000 people took us 18 months. And so we need to take the brown lines and the blue lines and shift them more to the left if we are to iterate and develop the kind of cadence that I think is needed to create the momentum to get us to a HIV vaccine regimen in the next couple of years. I think that's for us as clinical trialists to understand that these phase 1 trials have a lot of potential, a lot of interest, but we need to actually go through them and get the data and come up with a regimen in a plausible time for us to be able to test it for an effective HIV vaccine. So I'd like to thank everybody who works in this field, really, the hundreds of people involved in the HVTN, our funders and at AIDS, as well as the Bill and Melinda Gates Foundation, the site staff, the global CABs, and all the participants in our clinical trials program. It's been a pleasure to give you this overview, and I hope you found it useful.